click for IB Biology. Now we're moving into consider the form, by which I mean the shape and the function of proteins. We'll take a look at that. We'll look at some applications of shape and function as relates to parts of the course which you may not have covered, which you may or may not have covered so far. We'll talk about factors affecting proteins briefly, and then I want to bring us right up to date with a consideration of the use of technology in imaging proteins, which brings us very much into the 21st century and the exciting fields of artificial intelligence. So let's get started. If you want to make your own notes, there's a PDF in the description for you to annotate, and there'll be a quiz as well for you to check your understanding. Okay, so proteins largely, once they've adopted their shape through the folding from primary to secondary to tertiary, perhaps to quaternary, they overall, proteins tend to form either what we call globular shape or fibrous shapes. So your globular shapes are rounded or elliptical. They're kind of like lumps of protein. And if we look down here, we can see examples of that, these shapes like here, this kind of spheroid structure. Over here we've got insulin, which is a kind of lump of protein. And these tend to have functions such as hormones, or indeed proteins like this typically are enzymes, or they might be embedded in the cell membrane as channel proteins. Alternatively, the protein ends up being a long structure, which is where the descriptor of fibrous comes from, usually often composed of multiple helices held together by hydrogen bonds. Collagen itself is 3 helices, helicized together themselves, but then we have other molecules such as elastin, where the coiling actually imparts on the structure an elastic recoil, and you would find that protein elastin mentioned when it comes to blood vessels, so collagen in the outer walls of arteries and elastin in the muscle tissue, giving it the elastic recoil functions. So again, we see how biology starts at the molecular and then goes up to the physiological and functional level. Structure or shape tells us about the function. Here's some applications for you to consider, although I'm not going to go into these in great detail, but just reminders. You've probably completed a course on cell biology already and probably looked at membrane structures, so you'll recognize the diagram below with this large lump of protein embedded in the membrane, and likely it's acting as a channel protein. And so when we think about the amino acids within the chain, those which are in the outside of the cell or indeed the inside of the cell need to be hydrophilic, and so the amino acids with their R groups need to be polar, whereas when they are in contact with the fatty acid chains, they would need to be hydrophobic and non-polar chains in that region. The shape of the protein, of channel proteins, of course, will include the creation of the channel that runs through the entire structure, creating the possibility of facilitated diffusion. And then there's variations on this for thinking about our protein pumps in the cell membranes as well. We might like to think about applying the shape of proteins to the shape of enzyme molecules, and you'll recall, although we've not covered it so far in proteins, but the idea of active sites and the shape of the active site and how important that is in the mechanism by which enzymes catalyze reactions. Or we could look over here in my third example at immunoglobulins, or what people call antibodies. So these are protein molecules which have the capacity to recognize agents of infection, to lock onto them, and to destroy them. And so the shape of this molecule is critical to being able to identify the infective agent. Perhaps that's a pathogen or a toxic molecule. So shape is hugely important in determining function and maintaining function. And so the shape is determined, of course, by the primary structure, the secondary structure, the tertiary, perhaps the quaternary, very often the quaternary, and those all contribute to the shape of the molecule, which in turn provides it with its function. The functions of proteins can be disrupted by extreme conditions such as changes in pH or changes in temperature. With a change in pH, let's consider the idea that the peak of the curve here represents the most stable state of the protein. Now, it's important to understand that we're not just discussing enzymes. These are all proteins. So the protein is stable at this pH here, but if we decrease the pH, making it more acidic, or increase the pH, making it more alkali or basic, might be better, the addition of protons here causes a change in the shape, and a change in the shape changes the function. In this particular case, it's more than likely to make it less functional, which we call denatured. And that's equally the case when we create the alkaline conditions over here or remove protons. We end up changing the shape, change the function, and that's called denatured. So the argument here is that if shape determines function, then a change in shape will change the function. And it is probably the case that it becomes less functional. It doesn't work as well. And we would call that being denatured. This graph here is the effect of temperature. So as we increase the temperature, we increase the kinetic energy of the parts of the protein molecule. They vibrate more. And whilst the protein will tolerate temperatures up to a point where here it's very, very stable, but then we start to enter these conditions here with these temperatures, and the vibration of the molecule, the atoms and the structure starts to change the shape again, starts to change the shape, and this will lead to a change in function, and we would call that being denatured. You need only put the egg white into a frying pan to see what happens when we get to this part where we add so much kinetic energy that the protein molecule, the albumin, radically changes its shape and function by solidification. We call that denaturing. As a reminder, this is not the discussion on enzymes, which requires an additional component, and that will be dealt with in the enzyme videos. And finally, we move up to what I'd like to characterize as the 21st century. So Pauling, who I mentioned in earlier videos, used paper protractors and rulers in order to do his investigations and discoveries into the secondary structures in proteins. These days, we have protein databases which will present structures to you of proteins which have been determined by experimental work. Now, this experimental work is laborious, it's very time consuming, to determine the structure of the 100,000 human genes is likely to take an incredible amount of time to achieve. However, in the 21st century, we have moved from this level here, where we're simply looking at the ribosome and the location of protein structure, where proteins are synthesized. We can't see proteins in that image. We can simply see the ribosome organelle, to this level of detail in cryo-EM, cryogenic electron microscopy. Notice the size here at 7 nanometers, and what we're actually looking at is the shape of a protein molecule. So this is very much a 21st century development, which is being used in laboratories today. And then the remarkable work which has been done with AI, in which programs or algorithms such as AlphaFold, and it's actually now called AlphaFold 2, will actually assist the research scientist in predicting the structure of proteins, so that the idea of now characterizing all of human proteins is almost within our grasp, and huge progress has been made. And these two technologies are combined together. The predictive nature of AlphaFold is then connected to the cryogenic electron microscopy as evidence, and the two interplay with each other as they move forward, trying to predict the folding of proteins, their shape, and by definition, their function. This can be explored. This is the 21st century of biochemistry, and any serious student of biology and biochemistry really needs to take a look at these AlphaFold predicting databases, and I will produce some videos which will give you an introduction to how to do that, and perhaps your future will be in making breakthroughs in the function of proteins in the 21st century. The links below lead us to a subscription to the channel, and also to some of the playlists which we can use to further our studies of biochemistry.